From the American Neurological Association, I'm Adeline Goss. This is ANA Investigates. The glymphatic system was a neglected corner of neuroscience until recently. Now it's clear that the brain's system for the flow of fluid, nutrients, and waste plays an important role in sleep and neurologic disease. With glymphatics in the spotlight, researchers are coming up with innovative ways to study how this system slowly flows through the brain. Dr. Daniel Claassen researches the glymphatic system. He's a professor of neurology and chief at the Behavioral and Cognitive Neurology Division at Vanderbilt University. Dr. Claassen will talk about new directions in glymphatics research with Dr. Cheran Elangovan, assistant professor and Vascular Neurology Fellowship Program Director at the University of Tennessee Health Science Center. Here's Cheran.
Welcome to the show, Dr. Claassen. I really appreciate you joining us today for the podcast.
Thanks for having me. I look forward to talking about this.
Excellent. To begin with the interview, I would like to lay a foundation on this exciting field of glymphatic system. If you could give us an overview of what it is, and also could you tell us why a busy neurologist and a neuroscientist should stop and listen to our podcast?
You know, the last decade or so, maybe a little bit over a decade, there's been a new interest in how CSF, cerebrospinal fluid, is produced, how it flows, how it's cleared. You know, it appears that glymphatic flow, which is just, a term that's created that talks about glia and lymphatic movement of fluid in humans, it's probably related to CSF, but there's been a lot of interest in this, especially in animal models, because it seems to be organized. It seems to be regulated by aquaporin 4 channels. It probably is modulated in certain states like sleep, and it may have implications for important long-term chronic neurodegenerative diseases, especially as many of those neurodegenerative diseases are characterized by protein inclusions and proteinopathies, which are sometimes summarized as waste protein. So I think it's an interesting concept that in animals has been studied well. In humans, not so much, but it's getting better. But for the practicing neurologist, it's probably important to understand things that can modulate or be affected by that ultimately impact CSF flow. So I think that's probably why it's useful for a practicing neurologist to start thinking about.
It's definitely an exciting new field of research. And you mentioned sleep. Sleep has been recently more and more recognized as a key factor in brain health overall, especially in dementia. What do you believe are the most exciting research findings about glymphatics and sleep in the past decade? And, what does it tell us about the role of sleep in brain health?
Humans spend almost a third of their lives sleeping, and it's obviously critical for a number of different brain-related processes. And I think many pages have been spent writing about things like memory and inflammation and consolidation of thoughts and things like that. And so sleep, we know it's very important. As I said earlier, the CSF and how it flows is critical probably to a well-functioning brain. And we know in sleep, CSF flow changes. Generally, it's a little quicker. We see flow that has more pulsatile qualities. So I think when you think about neurodegenerative diseases in particular, most of them have sleep dysfunction. So, you think about Parkinson's, which has REM sleep behavior, Huntington's disease, very early changes to circadian rhythm, Alzheimer's disease, clear links between sleep dysfunction and amyloid deposition. So we know that sleep is probably important related to, and probably in effect modulates the neurodegenerative diseases. So I think understanding how sleep impacts CSF function and how potentially we could take care of patients who suffer from chronic sleep changes may have important implications for as we think of CSF flow and waste clearance and things like that.
Definitely. You mentioned finding out how sleep affects CSF flow. And along the lines, I know there's been extensive studies in animal models, but in human subjects, what are the techniques that you are using to research glymphatic function?
The best way I'd start answering that is just defining what we can look at using a non-invasive MRI. I think taking a step back, the regions of the brain that are probably important in glymphatic function have been established through different studies, including intrathecal gadolinium. And those studies seem to implicate that when you look at how, say, GAD contrast changes in the brain over, say, 3 or 4 days after intrathecal contrast. You see the contrast go up to the subarachnoid space and leave in the parasagittal dural space. So we know that that area is probably important for CSF egress, or even potentially a mediation between the extra CNS and CNS regions. So MRI can help us kind of identify those parts of the brain that are important in CSF flow and egress. And so we've looked at things like choroid plexus function. So the plexus obviously is the main part of the brain that makes a cerebrospinal fluid, probably is very important for neuroinflammation. So we've been able to establish methods to quantitate the volume of the choroid plexus, also look at perfusion of the choroid plexus using blood flow techniques. So MRI friends will be aware of arterial spin labeling techniques. And so we've been able to use those to quantify how much blood flow is going to the choroid plexus and how that relates to CSF production. We've also been looking at methods that you can quantify how fast and how much CSF goes through important parts of the brain. Recently, we've developed methods to quantify that parasagittal dural space. So that's the part of the brain that's right next to the superior sagittal sinus and kind of a superior parietal area, and is contiguous with some of the cranium areas. And we've also looked at things like arachnoid granulations, which are intravenous and probably also relate to CSF clearance. So we've taken the approach of looking at the quote-unquote glymphatic system by really focusing on how does the CSF get made, how does it flow, how does it communicate extradurally. And so using those methods, we've tried to understand how different diseases, and answering your question, how sleep may affect it. So the studies that we've done in sleep have been primarily in healthy participants who come in and they are sleep deprived. And then we have them take a nap during the MRI scanner. And we actually put EEGs on the participant whilst an MRI is being taken. We're able to record brainwave function, mark it for sleep, and at the same time get MRI information about the CSF flow and things like that to get assessments of how these systems and structures change during different sleep phases. So that's how we've generally tried to tackle a very difficult question, because it's hard to non-invasively assess these very slow-moving fluids like CSF in a person outside of an MRI scan, especially when they're awake and sleeping.
And I kind of read about your approach using DTI images as well. Could you elaborate a bit about that?
DTI measures how water flows. And so, one group of investigators tried to take advantage of some DTI methods to track fluid flow that was basically perpendicular to the lateral ventricle in an effort to delineate slow-moving fluid that may be glymphatic type fluid. So we read that interesting paper and tried to replicate it on some of our data. And it's an interesting idea. The concept is that, if you look at the perilateral ventricular area, you have some fiber pathways that traverse perpendicular, some that traverse orthogonal, and some that traverse kind of diagonally. And it's thought that the ones that are kind of straight off perpendicular to the lateral ventricles, those may not relate to white matter pathways, but maybe could be interpreted as diffusion flow that is of the lymphatic origin. And so you basically do a ratio of that region over the other ones that are white matter to try and guesstimate. So what we really found is that method is hard to conclusively relate to glymphatic clearance, and it's very related to age. So the older age of participants, the slower it is. It's also very related to white matter hyperintensities, as you'd imagine. So the more white matter hyperintensity you have, the slower it is. In the case of Parkinson's, it also seems to be related to the disease severity of Parkinson's disease. We speculated that it could be related to changes in glymphatic or white matter changes in Parkinson's disease, but that's been one attempt to try and take DTI data to make some inferences about how this system functions in neurodegeneration.
Thank you for elaborating on that. Do you think these techniques are capturing the glymphatic function completely, or do you anticipate any acceleration in different ways or techniques that will measure this going forward?
I think there's some very inherent challenges in trying to measure fluid that flows very slowly. I think imaging is pretty good at measuring CSF. So if you look at perivascular spaces or subarachnoid spaces, and even, bulk CSF movement, we're pretty good at that. And so whether or not those outcome measures are directly related to glymphatic function, I think that's a big question mark right now. I think there's some suggestions that it's probably related, but what this research is doing more than anything is it's really highlighting and emphasizing this important system in the brain that's, I think, been overlooked for a long time. And I'll give you a really great example of it, and that's Huntington's disease. So Huntington's disease has been a number of clinical trials where antisense oligomers are injected intrathecally into patients in an effort to reduce mutant Huntington protein transcription. In those studies, which started a number of years ago, one of the really interesting and unfortunate outcomes to several of those trials were increases in inflammatory markers like NFL, neurofilament light chain, and also ventricular size enlargement, and then clinically detriments in cognitive and motor function. And that's really kind of where we started asking questions about the CSF flow and putative glymphatic flow, is that if this is happening in experimental therapeutics, then we probably need to understand how the system changes in neurodegenerative disease or how it changes in over a lifespan before we start pursuing these very invasive treatments that may have unintended consequences. And one of the things that we've really found in Huntington's disease, and as you know, that's a fully penetrant genetic disease. And so you're able to look at patients early in onset, pre-onset, and during onset to get a sense of how these systems work. And what we found is that we see clear choroid plexus abnormalities even decades before disease onset. We see clear changes in CSF flow, especially during when motor symptoms start. And the velocity of that flow is very turbulent. And so it's an example of where the emphasis on understanding how CSF produce, how it flows, how it gets absorbed, those investigations, I think, will be very important for neurodegenerative disease, especially as it relates to future therapeutic interventions.
Thank you for elaborating on that. As far as Parkinson's disease, besides the fact that, we just know that abnormal protein clearance is affected, any recent research on Parkinson's disease and glymphatic system per se?
Yeah, Parkinson's is an interesting one because it's such a slow-moving disease. You think about your patients with Parkinson's disease, the disease is very hard to track because it moves so slowly. One of the big therapeutic interventions for Parkinson's disease is exercise. And I think, every exercise trial in Parkinson's disease works. Patients get better. They feel better. They move better. They think better. Given the knowledge of how exercise, cerebrospinal fluid flow, putative glymphatic function, how those are related, we actually have started to look at that. And in that study, we took patients who were naive to exercise, got into a community exercise class. A big thing in Parkinson's is rock steady boxing, and followed them for just over 3 months and then scanned them at baseline and follow-up. And what was really interesting is that patients, when they did the exercise classes, had improvements in motor function, improvements in processing speed, and we saw changes in important markers of CSF flow, namely choroid plexus function and CSF. So we think that, exercise could be an important treatment that might work through improving CSF flow and putatively reducing things like waste protein, which probably are important in the disease process. One of the really fascinating things of that study, which was not anticipated and has led us on a new series of investigations, is the relationship of intravenous arachnoid granulations and disease severity. So when you look at patients that have worse motor disease, they have much greater volume and size of these intravenous arachnoid granulations, which we know are important areas for CSF clearance. And so that interesting discovery really is encouraging us to continue looking at how these things change over time and how we can potentially develop treatments to modulate CSF flow and clearance to potentially help patients reduce the effects of disease, but also be potentially disease modifying.
Besides exercise, where do you think is the current research leading us in terms of strategies or therapies to improve glymphatic function or CSF flow?
Yeah, I think non-pharmacologically, the most obvious things to talk about are sleep and exercise. I think the other part that is, not clear is therapeutics, and would certain medications, improve these processes. And there's been some suggestions of this. If you look at certain anesthesia compounds, they might potentially modulate this process. And so you could imagine investigations into therapies that could potentially target these pathways to improve CSF flow and function. And that could be an emerging area for disease modification and treatment. And I think those investigations will be very interesting to look for in the next decade.
Absolutely. These are exciting directions, and I'm going to be really looking forward to these developments in the future. Coming to the end of the podcast, what do you see as the most important direction for future research into the glymphatic system and CSF flow? And do you have any advice for researchers in this field or those who may be starting out into a research pathway?
Yeah, I think replication is really important in this field. I think if we've learned anything in the last 5 years about the state of neurodegenerative research, we have to replicate findings. And I think looking at single center studies, like our own, it's really important for other folks to replicate that to see if they say the same effects. That's really critical if we're going to move this forward. I think the other is understand the assumptions of the model. We use words like waste protein clearance and things like that when, in fact, we may be talking about important areas like inflammation. And so I think developing models that really are based on hypothesis-driven experiments, I think, are very important in this field as we coin these terms that we throw around a lot, but they may be different. So for young investigators, I'd say, you can do a lot by replicating data. I think you can really advance the field by doing a study that may not be the most novel, so to speak, but you learn methods and you learn to see how these data hold up when you do it. I think that's a valuable part of learning how to build your scientific program and thoughts. Mentorship and open science, I think, is also very important. In our group, we've really tried to make these tools for MRI analysis open, so anyone can use them. And I think that helps the field. And so encouraging open data and open science to make sure it's available to everyone, I think, is important. So I'd encourage that mindset as we go forward.
These are some great advice for newcomers and those who are already looking into this. And I really want to thank you for taking time to join our podcast and giving your expertise and sharing your thoughts on this developing field. Thank you.
Yeah, thanks for having me and thanks for the conversation.
That was Dr. Daniel Claassen of Vanderbilt University, interviewed by Dr. Cheran Elangovan of the University of Tennessee Health Science Center. ANA Investigates is the official podcast of the American Neurological Association. The show's producers are members of the ANA Education Innovation Committee, chaired by Michelle Johansson. I'm Adeline Goss, podcast host and executive producer. Our program was produced today by Cheran Elangovan, Caitlin White, and me, with help from Jen Hurley. You can listen to past episodes of the show, subscribe to us so you never miss an episode, and access all of the ANA's educational programs on the Education tab at myana.org. That's it for ANA Investigates. See you next month.